growth of fungi. And when you are doing this process, it is better that you keep a control. When you do this process, it is better that you keep a control in order to find out whether the growth is from the tissue or it is not a contaminant, because fungi do occur as contaminants, and we should be very careful when you are handling these fungi, isolating these fungi, because we should not end up with a contaminant saying it is an endophytic fungi. So this picture depicts the steps involved in the isolation of the fungi, where you collect the sample, root, stem, leaf, then pass it on to a series of sterilization procedures where you use sodium hypochlorite followed by ethanol, and washing the tissue in sterile distilled water. Cutting the cut portion is placed on the agar, and it is called as an agar impringement method, and the plate is being sealed and kept for 48 to 72 hours for growth of the fungi. So after the growth of the fungi, the organism can be identified by using lactophenol cotton blue staining method, and once it is identified, it can be subcultured onto sabro-dextrose agar, and it is preferable that we use a medium called as a potato dextrose agar.
Hello, sir. Yeah. Your screen is not visible.
Okay. I'm sorry. It has got stuck up. Now is it okay, sir?
No, sir. Still it's unusable.
Oh. Now it is okay?
Yes, sir.
Yeah,
yes,
sorry.
sir.
I'm sorry because there was some advertisements coming in between, so I accidentally clicked that. Okay, we'll go to the presentation mode. I'm sorry for the inconvenience. So we are looking into the different medias that are used in the isolation of the fungi. It can be a potato dextrose agar or a malt extract agar or a sabro-dextrose agar. But whatever media you use, we follow a method called as a solid impringement method, by which the tissue is being placed on the agar for the growth of the fungi. So this is just to show you how an endophytic fungi occurs on a media, right? On the top side, you can see the fragments of leaf, root, seed, and stem that are being cut. And here beneath, you can see the picture. I hope the picture is visible. The picture is visible, sir? The slide
yes, sir,
is, yes. Yeah. Thank you. So this is how the endophytic fungi grows. Apart from this, we also keep what is called as a control plate. If there is growth in the control plate, the entire process has to be repeated, so that we have to be very sure that we isolate only the organism, that is the endophytes, and not the fungal contaminants. Right. Just to show you a picture of what are the types of fungi, and you can see Aspergillus flavus. You can see the colony morphology followed by the microscopic appearance. And here you can see the Aspergillus niger, the colony appearance along with the microscopic appearance. And here you have what you call it as a dermatitious fungi. We call them as Curvuraria species. And here you can see the microscopic appearance. And here you have one more fungi, which we call it as Aspergillus nidulans. And you can see the morphological microscopic appearance. All these fungi are identified by conventional method, namely by lactophenol cotton blue staining by microscopy. And apart from this, you also have different methods of identification. On certain fungi which do not sporulate, we go in for a molecular method of identification of fungi. And this is the macroscopic and microscopic feature of the isolated endophytic fungi in my laboratory. So here you can see in the picture, all the entire growth is a pure growth, and you don't get a contaminant. Once the fungal grows in the segment, the fungi will be transferred onto potato dextrose agar plate or sabro-dextrose agar to note down the colony morphology on the front as well as on the reverse side. Once the fungi has grown over the plate, then a wet mount is made to identify the fungi. And this is how we go about identifying the endophytic fungi. When you talk about, I've already made a mention about this. If certain fungi which do not sporulate are taken for molecular characterization to identify them by using ITS region sequencing. This region is a conserved sequence among all the fungi. As a result, this unknown fungi can be identified by using this ITS sequencing. And when you talk about once the fungi is been isolated, the fungi is subjected to what is called as a primary screening, where we identify whether what sort of a compound is being produced. And this is how, right. Okay. We first try to take the fungi and we make a culture onto the medium and to analyze the production of bioactive compounds. And there are two methods that are followed, namely solid state fermentation and submerged fermentation. In solid state fermentation, we use metabolites like solid substrates like rice bran, coconut oil, vegetable waste, or a husk or an orange peel. Whereas in the case of a submerged fermentation, we normally suspend the fungi in any broth medium and we kept it in the shaker for it to grow for nearly to 15 to 20 days, during which you get a mycelial growth. And this fungi, when it grows in the medium, it starts producing the bioactive metabolites. As a result, these metabolites can be further recovered by the recovery step. This picture shows you the difference between a solid state fermentation and a submerged fermentation. This is just to show you an activity when you are doing this work. Once the fungi is been separated, after the end of the 15 to 20 days, the fungi will be taken from the shaker and it will be, the crude extract will be tested for the activity against the clinical isolates. So if there is a production of an zone like this, then it will be taken for further process by using various solvents. The bioactive compounds will be extracted. So when you talk about the production of these bioactive metabolites, once the main important point in the culturing of the media is optimization of the culture conditions, where we optimize the carbon source, nitrogen source, the inoculum that has been produced, inoculum that has been used, the incubation period, temperature, pH, everything. This is very important because based on the bioactive metabolite, what we are looking for, we need to have a suitable carbon and nitrogen source. As a result, we will end up in the compound which we require for our studies. And this is the various medias that are used for production of bioactive compounds. And these are the different medias and the different conditions. However, there are a lot of medias that are being there, but I've just made a mention about only few of them and the conditions that are required. Then once the broth, the fungi has been grown, the mycelial mat is separated and the broth is being subjected to ethyl acetate fractionation, where you can see on this picture on the left side, this is a growth of Chaetomium species strain on malt extract broth. Since we have kept it on a shaker, you can see the fungal balls appears very round and it looks very perfect. Once this step is obtained, the entire culture is being taken for ethyl acetate fractionation, where we using a solvent, we add to the broth. We first separate the mycelial balls and the culture filtrate. And to the culture filtrate, we add what is called as an ethyl acetate or any solvent, or depending upon the literature, depending upon the one that is shown in the literature. And once it is kept in a separating funnel and allowed to stand for a few minutes, you can see two different phases that are appeared, and the topmost phase will be removed and the lowermost phase will be taken for the activity of bioactive compounds. So during this process, once the material is taken, the crude culture filtrate, then it will be kept in a desiccator and it will be dried and using a solvent, the crude extract will be dissolved, and it will be taken for GCMS studies for structural elucidation. First, to find out what are the compounds that are present, and further that is followed by the purification step. So when you talk about purification, that it includes filtration. There are a lot of procedures are involved. Only after putting a column and purifying the compound, we'll be able to say which fraction is able to give the result, and so that we can take that fraction and go for the crystallization of the compound and doing NMR studies. So these are the various solvents that are used for the recovery of endophytes. Recovery of endophytic derived bioactive compounds. So depending upon the compound which you look for, the solvent varies. And that is a major important thing in this research. And when you talk about, many people would be thinking that why these endophytic fungi are so much important, right? Looking into the literature, we have come across some interesting compounds produced by these endophytic fungi, which includes the famous drug Taxol, camphothecin and vincysteine and vinblastine. And these have been reported, they have been extracted from an endophyte which have been grown in Traxus bififolia. And you have a huge list of host plant which are giving shelter to these endophytes for the production of bioactive metabolites, and which are of very great importance. Apart from this, I have already told you that apart from the land plants, apart from the seaweeds, even endophytes have been reported in mangroves, and we have come across, we have a huge collection of endophytes that are isolated from even from mangrove regions. So these mangroves in Tamil, we call them as Sadupu Nila Kadugal, where these mangroves are living in highly saline environment, and they have a novel mechanism. And as a result, the endophytes, the plant as well as the endophytes are explored for their bioactive metabolites. So this is a picture, this is a tabular column, shows the endophytic fungi that has been reported in seaweeds. So seaweeds that are located near the Gulf of Mannar and a lot of areas around the sea region, sea belt, they are also a rich source of these endophytic fungi. And our laboratory, we are working on the seaweeds and the land plants for the isolation of endophytic fungi, right? So before moving on to the things, I would like to thank the organizers, and I'm very sorry for the.
Thank you so much, sir.
The inconvenience, because it's been a long time we have been on online and usually go for an on an offline presentation. If there is any question, I'm ready to answer for that.
Thank you very much, sir, for your great information regarding antimicrobial resistance and endophytic fungi. It will be definitely help us in future. Thank you so much.
Thank you, ma'am. Thank you for the nice introduction, and thank you very much for the organizers for giving such a nice opportunity to be from down south, Tamil Nadu, to see you all in Maharashtra. So I have gone in 2 occasions to Maharashtra. Thank you. Thank you very much. Thank you.
Thank you very much. Now again, session is open for question and answer. I kindly request you all participants to propose your questions in front of experts. I request all the participants, if you have any question regarding this, then ask. Okay. Now I request chairperson of the session, Honorable Professor Dr. V.M. Ghorpade, Dean PG Studies, Yashwantrao Chauhan Institute of Science, Satara, to chair the session with his valuable words. I request Dr. Ghorpade.
Actually, ma'am, actually there is some problem in SIRS network, so they are unable to connect with us.
Okay, we will wait for 5 minutes. I request all the participants to stay online. I think there is a network issue. I think there is huge network issue is there. And due to that, now I would like to call Mr. A.S. Bansode sir for giving vote of thanks.
Am I audible?
Yes, sir, you are.
After 2 sessions, I would like to thank most inspirational personality, our chief guest of this session, Dr. D. Arvind Prasann, Associate Professor, School of Bioscience, Periyar University, Salem, for giving us and sharing our knowledge with delivering a talk on antimicrobial resistant epiphytic fungi. And sir, it's our pleasure to hear you. Also, I thank our chairperson, Honorable Dr. V.M. Gorpade sir, Dean PG Studies, YCIS Satara. Okay. Thank you for all.
Thank you so much, sir. Now session is over here. We move towards our valedictory function.
Excuse me, ma'am. At what
Yes.
time shall I join online?
Now it's now valedictory function is start
Okay, sir.
on the
Okay,
same
ma'am.
link.
Okay. I'll join in another device.
Yes. No, sir. Yes.
Yeah.
We continue here.
Yeah. Okay, you
Okay,
can continue,
okay.
but I'll join in another device.
Okay, sir. Okay, sir.
Yeah.
Okay.
Thank you. Thank you. Thank you.
Validity function is starts immediately within a few seconds. Sorry, sir. Oral presentations are there, and due to that, validity function is at 5 p.m. Hello, sir. Hello, sir. Am I audible to you? Hello, sir. Join. Another presentation. Hello, everyone. Hello. Am I audible to you all? Hello.
Yes, ma'am.
Okay. Our oral presentations are start already on another link. Can you please join this link? I will provide you this link on WhatsApp again. Please join this link. And validity function also at 5 p.m. Can you please join the link? I will send you the link of that oral presentation.
Excuse me, ma'am.
Hello, sir.
Can I join at 5 o'clock for the valedictory?
Yes. Yes, sir. Validity function is at 5 p.m. But
Okay, I'll join.
oral presentations are started.
Okay, okay. I will join at 5 o'clock for the valedictory.
Okay, okay, sir. Okay, sir.
Yeah.
Okay, no problem,
Okay.
sir.
Thank
Okay.
you.
Thank you, sir.
Thank you.
Thank you very
Thank
much.
you. Thank you.
I request all another participants. Can you please join the WhatsApp link? Sorry, link provided on the.
Hello.